Hello, everyone, for another session of cell migration seminars. Sadly, we're not going to have Dave today. He's unwell, but hopefully we can pull him for some other day. But first, we are going to have Professor Ben Goult today. Ben is a biochemist and structural mechanobiologist, and since 2005, Ben has been working on protein tailing. So, well, that's a long time working on tailing, and its role in coupling integrins to actin and microtubule cytoskeletal at the core of integrin-mediated cell adhesions. So he did his PhD at University of Manchester, and after a brief spell in industry, Ben moved back to academia in 2006 to do a postdoc with David Critchley to work on structure of talin. Since 2014, Ben has been in University of Kent to set up his own research lab looking at mechanobiology of talin. And the main focus of his lab has been how the rod domains and talin act as force-dependent binary switches. And I think that's what he's going to talk about today. Thank you so much, Ben, for accepting our invitation today. We're really looking forward to your talk.
Great, thank you, Ankita. Thank you, Jen, for the nice introduction. I know I helped write it myself, but it was still very nice. And thank you, everyone, for listening and the opportunity to present to you all in this excellent seminar series. Today, I want to talk about our ongoing saga to try and understand this protein talin and its function in all integrin adhesion complexes, essentially, almost all. And more recently, we've started to really develop this idea of talin's role in mechanical memory and how these binary switches, which Ankita introduced, provide a way to maybe store and capture information in the shape of these molecules. So that's what I want to talk about today, to try and introduce this concept of spatial organization, which can be encoded by these binary switches. And I'll just say I've spent the afternoon trying to trim this down to 25 minutes. So the opportunity to go slightly beyond that is quite a relief. So that's why I'm actually not talking quite as fast as normal. So this is the protein talin. And in this audience, we don't need much of an introduction in what talin does. And we're all probably all familiar with integrin adhesion complexes. Needless to say that the cells of our bodies have got integrin receptors on their outside, which can physically grab onto the extracellular matrix. And in doing so, the cell can adhere, or it can migrate or move. And just inside the cell, the protein talin can pull on these short cytoplasmic tail of the integrins and coordinate the activation state of the integrin, holding it in a high affinity state. But what talin also does is it couples it to the cytoskeleton, both to the actin and also indirectly to the microtubule cytoskeletons, serving as a connection between the integrin ECM complexes and the cytoskeleton. And the classical view of mechanosensing of these systems is that when talin experiences forces, both from intracellularly or extracellularly, it can undergo these changes in its shape and recruit the protein called vinculin. And vinculin is an adapter which binds to talin and couples it to the active cytoskeleton, reinforcing these linkages. But what's interesting is here I'm defining four proteins: an integrin, a talin, and an actin with vinculin cross-linking it. But onto this simple core complex of these adhesion scaffolds, you can assemble many different diverse structures. And these are all mediated by this integrin talin actin core from these very dynamic adhesions in a migrating cell, as shown in this fibroblast crawling along the surface in this beautiful video from Mark Morgan, to these more long-lived persistent junctions in adhesions in these myotendinous junctions connecting these muscle cells together. Talin is also essential in podosomes and invadopodia, and in the coupling of the cell division and the adhesion machinery, as shown by this cell at the bottom undergoing mitosis, letting go of 99% of its adhesions, and then immediately undergoing mitosis and then immediately respreading. And also what we're interested in is these synaptic adhesions, which are essential for these intimate cell-cell junctions, which enable signaling between adjacent cells. And talin is essential for all of these processes. And as Ankita pointed out, I've been working on this since 2006 when I joined Dave Critchley's lab. I was actually working on talin in 2005 because when I was at AstraZeneca, I was asked to test a load of NMR software. and choose which one they were going to adopt. And at the time, the state-of-the-art one was the scripts were being written for it by a guy called Igor Barsukov, who was worked with Dave and was my boss for a few years. And he sent me some scripts, and he sent me Talin data, model data to work on. And that went really nicely, leading to me to go down to Leicester and ultimately to work on this protein. And so for the last 10 years, we've known what this molecule looks like. It looks like this. And it literally is 10 years, because when I was looking, it was last week that we published this paper and defined that there's 13 switch domains in Talin. And these are now called R1 to R13. If you look at papers prior to this one, then the naming of the domains of Talin is a hot mess. But this clarified that. So now they'll just be referred to as R1 to R13. And in this simplest arrangement, the N-terminal head domain binds to integrins, the C-terminal regions bind to actin, and that positions all of these helical bundles in this force transmission pathway. And something which we noticed in 2013, which dictated the flow of my research ever since, was that Talin changes partners in response to force-induced conformational change. And that's the crux of what I want to talk about today. And it's illustrated quite nicely, I think, by this video showing a Talin molecule here, which our initial switch we discovered was this R3 domain, which when it's in this closed compact form binds to this RAP1 effector called RIAM. But then when force is exerted on this molecule, that domain unfolds and simultaneously exposes two vinculin binding sites. So you can see that happening here. We pull on this molecule, you cause this transition and you change this signaling. Something which I've not got time to talk about today, although it maybe turns out I would have had time to talk about today, is that this stretching of Talin can activate vinculin. It's shown in this video, but this actually happens. You stretch a Talin and you can relieve autoinhibition of vinculin. But what I was interested in when I was in Dave's lab and what I set up my lab to answer was how much force is required to unfold the R3 domain to cause this conversion of mechanical force into a biological signal. I just want to take a brief sidestep just to quickly acknowledge Sam Barnett, because over the last year, we've been working a lot to model these structures and to actually draw them and visualize them. And he was central to making these beautiful videos, which I'm going to show, which I think are very instructive of what we're talking about. But the question was, how much force is required to open that R3 domain of Talin to cause that change in biological signaling? So I emailed Mike Sheets in Singapore, and he put me in touch with a professor there called Yan Ji in the Mechanobiology Institute. And ever since that first email, we've become really good friends, and we've really done some, I think what is cool, you'll see what you guys think, but to try and work out what's going on here. And the initial experiment which we did was to take 3 of these domains of Talin and attach them to the cover slip of a microscope, which then we, via the N-terminus, and then via the C-terminus, we could attach that to a magnetic bead, which then allows us to use a permanent magnet on a magnetic tweezer deck to pull on that bead and exert physiologically relevant forces onto these molecules. And when we do that, when we stretch these 3 domains of Talin, what you see is that as the force increases, do you see my cursor?
Yeah, I do.
You do?
It's tiny,
okay,
but yeah, I do see it.
As the forces on the molecule increases, this molecule gets pulled taut. But then what you see is these large jumps in extension. And there's 3 of these. These are these Talin rod domains unfolding independently. And I would have had time to show you, but the R3 domain has got this, is the initial mechanosensor, is this domain here. And we can show that's the case because we can make mutations in R3 which stabilize it and shift this initial folding step. So this defines all 3 of these domains as switches because they open and then they close when force goes away. This is 6 cycles of the same thing, stretching it to spaghetti and then letting go, and it immediately resets itself. And then we wanted to expand this to look at the whole of these. Having found 3 switches, we wanted to see what all 13 of these look like and to look at the complete mechanical response of this molecule. And when we stretch the 13 domains of the Talin rod, we get this panel here. And this is still mind-blowing to me because it's, even though we've had it 7 or 8 years, that it looks, it's this complicated, but also this robust. And what you see when you start to increase force is you see R3 unfolding first. That is the initial mechanosensitive responsive Talin. But then what you start to see is this step-like behavior as these domains reach their force threshold and unfold independently. So this shows that each rod domain is a mechanical binary switch, and each step corresponds to a single domain converting from a folded state to an unfolded state. And because we have ligands for most of these in the folded and unfolded states, each of these steps represents a domain mechanosensing and changing its biological signaling in response to force. Another hand up. Do we answer questions as we go? Or
yeah, would you mind? I think Claire has a question, if that's okay.
Hello,
I just
Claire.
wondered if those. Hi, Ben. Great to see you. I just wonder if those go in a similar, specific order that unfolding.
Yes. No, it's a good question, and sort of. So these ones in this R3 always goes first. These ones about 8 to 10, 8 to 12 piconewtons. It'll always be one of those goes second. And these really stable ones are always really stable. So within the order which it unpacks, there's a bit of stochasticity to which one of those in this pulling end to end. But obviously, when other things decorate this and different forces load on it, then they will, I think they'll be in a very specific order. So they are in an order, but you couldn't tell which of those three would go next. It'd be one of those three. That answers the question. So this leads to a framework we've been trying to develop to understand mechanotransduction and mechanical signaling through these integrin adhesion complexes, and this notion of Talin serving as a mechanosensitive signaling hub, and where it can recruit different molecules depending on the status and the shape of that individual molecule. So here I'm showing a low force condition where all of these domains are folded, and you recruit a certain subset of signals, and there's a binary pattern of 4 folded domains, and the readout is this assembly of these molecules which decorate. And then if there's a contractility event, a signal comes in, switches on the motor proteins, that can pull on this protein, and then the weakest domain in that order will unfold. It will displace the signals on the outside, but recruit things like vinculin, which reinforce these networks. And then a subsequent contraction will change that and update it again. And the readout will be these altering signaling complexes which form. And this led to, when I looked again, this is how quickly time flies. This I thought was last year, but it was actually Christmas of 2021. We started to put all this together into one of these, my favorite series of review articles, An At a Glance in Journal of Cell Science, where you try and conceptualize the whole idea into a poster. And this is a blatant sales plug, but if anyone would like a copy of these, we got sent loads of paper prints. I'd be happy to send them out, and just drop me an email. But on this poster, we started to summarize all of the different ligands which we've got, which are interacting with this. And this is an abridged list because these are the ones which are published. But what you can start to see is each of these domains is switching on and off different functions as a function of its shape. And because you've got this massive network of competitive interactions combined with the switching of 13 binary switches, this is really complicated. And it's even more complicated because there's lots of different ways that this molecule is getting pushed and pulled via its linkages to the actin cytoskeleton and to the microtubule cytoskeleton and the extracellular environment, the stiffness, whether the cell's migrating. But this is incredibly robust because development happens nearly perfectly. So these mechanical signals and these protein and chemical signals all integrate in a really reproducible signaling way. So there must be, it has to be metastable in response to all of these diverse multiple inputs. So what we want to understand is this coding, or this mechanical coding at the protein level. And more recently, I've changed the naming of this ever so slightly because everything I've spoken about so far is on a single Talin molecule. And obviously a single Talin molecule doesn't exist in isolation. It exists as part of a complex meshwork of molecules such as Talin, such as vinculin, which assemble on these integrin complexes and make a meshwork of mechanical switches, which is why I called it a meshwork code. I thought it was more of a mesh code, was more of an apt name to be more broad than just a pure Talin code. But then as a function of all of the inputs, and when this cell reaches mechanical homeostasis, then there'll be a certain pattern of these switches which are then decorated with signaling molecules in a context-dependent manner, depending which binding sites are accessible and available, to give rise to very specific signaling complexes. And that's the idea we're trying to develop and trying to prove. And if you imagine a cell in culture, these adhesive structures, these cytoskeletal structures, they're very dynamic, and they change over the tens of seconds because the cell tends to adopt a migratory phenotype. So when you imagine what's happening to those Talin switches, then you've essentially got a balance between two changing forces. You've got the extracellular forces as this cell's migrating and moving, which are constantly changing. And then you've got the cell's internal cytoskeletal force generation machinery switching on its motor proteins. So the resulting switching pattern, which you end up with, is the sum of those to give rise to quite a dynamic switch patterns. When I was coming back from Australia in 2019, sat at the airport, I started to think about a question from Peter Gunning in Sydney, and he asked about how all these Talins could be so robust. And something which occurred to me, and is a very simple question, is what would happen if the extracellular environment is constant and predictable in this scenario? And by this, I don't mean it never changes, because the cells invest a huge amount of resources into remodeling their extracellular matrix to make it completely predictable. They know what it is because they've essentially built it with the help of fibroblasts and other players, obviously, but they know what that is. In this scenario, this equation gets much more straightforward, because now the extracellular forces are not changing as much, or they're known. So it's no longer a tug of war. Now what we could imagine is that you could have a series of switches which are being operated via the cell's force generation machinery. So the cell could receive a signal, switch on contractility, and pull on this and update this pattern and update the resultant signaling. And that was the general idea which led to this paper, where I started to try and work about that if these systems are ordered and if these adhesions are more ordered than what we currently envisage, then they can be able to store information. You could write information physically into them, because you could change the shape of these molecules using small changes in contractility. And in doing so, you could update the signaling of that cell, but in a quantized way, written in a binary format. So that was an idea I had. I wrote an article on this. This was under review for, I don't know, 9 months or something. It took ages. But it really sharpened it up. And in that paper, what I tried to argue, after the first half was about the actual serious stuff, and then the second half was a bit more what this might mean if it was ordered. And one of the ideas that it led to was this notion that maybe you could have a binary coding on both the pre and the post-synaptic side of this, which would alter the activity of that synapse, but also be information that is written in there. But the obvious question, which everyone asked and kept me up at night, was how, though? That all sounds nice, and well, sounds well, it depends what you think. You might not think it's nice, you might think it's horseshit, but I mean, it sounds like out there. And one of the things we started to do was we actually tried to build a simulation of Talin receiving signals, updating. And we suddenly realized that was really hard. But one of the things we realized as we started to build the components to feed into that is that there's something which we had completely overlooked in terms of the dimensionality of these systems. And that's what, if you look at what happens if you draw these molecules to scale, this is a scale drawing of Talin with all 13 domains folded. And the rod's about 55 nanometers. If we just take 2 of these domains and draw them out like this with 2 ligands bound to them, and then watch what happens when one of these switches opens up, it introduces 50 nanometers in length into this molecule. So in essence, a single domain opening is pretty much the same length as the entire Talin rod. So the rod's doubled in length with just a single domain opening. And if you look at the spatial organization of those molecules which are sat on it with these hypothetical orange and blue and pink shapes, then they've been relocated dramatically as a function of that single switch opening. So you've obviously, with the switch which is opened, you've got the change in binding partners. But perhaps more interesting is the cyan guy, who is still bound to the same site he was before. He's just 55 nanometers further away from where he was before this unfolding event. And when you draw that, then what you can start to see is that this molecule is undergoing dramatic changes in its dimensions from a compact 86 nanometer long molecule, unpacking these domains and going in quantized 40 to 50 nanometer steps in its length. And it's 40 to 50 nanometer relocating of these binding sites for these different proteins. So what we came to realize is that there's a massive distortion in how we usually draw and think of these complexes. And these two drawings here are incredibly useful. They've shaped the field, and they're really, and I'm not just saying that because Claire's here, it's actually because it's true. But this is like a framework where we can understand this, but it's only a snapshot of a Talin, and it doesn't capture the range which is seen in the supplementary figure of this. And I'm even more guilty here because I'm actually showing the switches change shape, but no change in dimensions. So I linked this to the Mercator projection map of the Earth, where this is incredibly useful. This is a common story and a common idea which everyone can share and understand. But it's distorted in that it, like in the Mercator projection map, where Greenland's the same size as Africa, which obviously it's not. But it means that you can use it, but you lose a key part of the information by reducing it to this single image which everyone holds. If we re-envisage that and think about this drawn to scale, so here's those scale drawings of Talin, 5 different Talins in different states, but here put the ligands back onto it, then what we can start to see is that as these molecules, as these Talins unpack these switches, it's spatially organizing molecules in the Z dimension. And by Z, I mean, I'm calling X and Y the membrane, and this is coming down away from it as these switches unfold. And I've just stressed here that this is only 3 switches shown unfolding, because then it goes off the page. Hello, Claire.
So Talin is at a 15-degree angle relative to the membrane.
Only in
a cell.
Under the nucleus, I don't think it's a 15 degrees.
Yeah, I think it is, because the actin cytoskeleton is cortical, and it doesn't pull into the middle of the cell. It pulls tangential to the membrane. So
yeah, but not if you've got all of your attachments at one end of like in a synapse or in a thing where it's at one end and then you're pulling.
Synapse, yes, but in a focal adhesion, I said synapse maybe, but focal adhesion, it's always going to be very close to the membrane, even when it's extended. The actin cytoskeleton, the cortical actin cytoskeleton isn't further than 100 or so nanometers from the membranes. It's not pulling in normal to the membrane in a focal adhesion.
The angle bit's kind of important, but it is the same principle as stands. You're still going to position these along an angle. It doesn't matter
absolutely
which way that line is.
Yes, but it's very close to the membrane. It's always going to be close to the membrane. I think that's the main point that I wanted to. If it's a focal adhesion, it's going to be basically normal, being extended along the membrane, because that's the force vector. It's not into the middle of the cell.
Yeah, well, maybe we should chat offline, because I think the interaction of the localization of where Kant goes around these and the lengths, how far it can reach, indicates that it's going further into the cell than just a straight up 15 degrees, because you physically localize these molecules to the edge of these adhesions as a function when it binds, when they get stretched out. So I don't think it's coming down at 90 degrees vertically up, but I also think that 15 degrees is only one fringe case of it.
Okay.
And we have a question in the chat that seems potentially relevant to this discussion. It was from a few slides back when you first showed the different dimensions of the pulled out Talin. And so Aaron Cram, it says, very cool. Does the molecule really extend like that when it's embedded in the actin cytoskeleton? So I think that's relevant to what you and Claire were just discussing.
And I think so I'll show an image of Talin in dendritic spines later on, but I think the best evidence for it is the work from Mike Sheets with the Felix Margadan cyclical stretching of Talin, where they could show Talin was stretching from 50 to 350 nanometers in cycles by measuring the end-to-end distance. So that to me shows that between 6 and 7 domains are opening up a Talin during that duration. And in a migrating cell, then that that's limited because the lifetime of the linkages, because then they disassemble at a certain point, so they turn over. I'm actually not sure if that answers the question now. What was the question again? If it actually does that in the cortex.
Right. When you have, since you
Sorry.
have the, the actin cytoskeleton there,
Yeah.
can they actually extend that far?
Yeah, well, that's measured from, the end-to-end distance of the Talin would indicate that they do stretch, maybe not to 350 nanometers easily, yeah.
Great. We'll let you
Thank
continue
you.
on, and people can keep dropping
Yeah,
questions
please
in the
keep
chat.
it. I'm enjoying this. I forgot where I am now.
Thanks.
Okay. Oh yeah, so these are proteins are spatially organized in Z, whatever angle you've got of your Talin, they're still spatially organized along a force vector. And just to highlight, I'm not going to go through everything we've done, but just one example of an enzyme which is organized in this way. And that's the cyclin-dependent kinase 1. And this is some work done with Matt Jones and Martin Humphreys in Manchester, and with help from Tom Sashchenko, and then some single molecule work with Yan Ji's team. And CDK1 is a Talin ligand which actually sits on Talin on the R8 switch. But the interesting thing is it phosphorylates lots and lots of adhesion proteins, and I'm sure Claire may well put her hand up again in a second, but it phosphorylates lots of things around this site. But it also phosphorylates, one of the substrates is actually a feedback mechanism where it phosphorylates onto Talin. So Talin physically positions CDK1 at specific layers within this cellular structure up until CDK1 phosphorylates Talin. And then you get this dramatic shift in the stability of the domains which CDK1's bound to. And that's another opportunity to show a video to try and visualize this, where CDK1 binds to a switch. So it phosphorylates things in this vicinity. But by destabilizing that domain, it ultimately introduces over 100 nanometers extension into the angle, into the length of the molecule along whichever angle that's at. And this, to me, it indicates not only can you write information into these in the form of signaling outputs, but that you can also have, I don't know what happened there, there's possibility of kind of essentially a logic gate, because here you've got, if you've got tension, then and an active kinase, you get this 110 nanometer extension. If you've just got tension, then the next weakest domain will unfold. Or if you've got an active kinase but no tension, it can phosphorylate lots of adhesive substrates. So here's an example of an enzyme being specifically located at certain layers within a focal adhesion or within an adhesion structure as a function of these switch patterns. And what's not hard to imagine is that these molecules being spatially organized, we've just drawn a membrane-bound one in orange and then a green and a pinky blue one, that it's almost like a Venn diagram. In some of these patterns, you bring these molecules really close together. And if you have an enzyme and a substrate and you physically hold them close together, then you can have high activity. If you physically hold them far apart, then you can have low activity because they can't reach each other. And if they're freely diffusing, then you get the classic diffusing kinetics, which we all kind of know and love. And this idea of scaffold proteins coordinating enzymes isn't new. If anyone who's done a cancer biology or a cell signaling undergraduate module will have talked about the MAP kinase pathway and the kinase suppressor of RAS, this predominantly unstructured protein which binds all of the kinases in the MAP kinase cascade, bringing them together in space and time to control the flow of information. The big difference here is that Talin is not an inert molecule which is just flopping around to do that. Talin is mechanically operated, so it can move these back and forwards relative to each other, would be the idea. So the next question we're interested in, that these spheres on this figure were just drawn as 30 nanometer circles because they look nice and it's kind of aesthetically pleasing at that size. But what actually determines the size of those zones of activity and when and where these this Venn diagram will intersect? And what's this one? I've just put some slides back in, so I've gone out of order. Yeah. So then the next thing we realized was that this molecule is changing shape on these large scales. Things like CDK1, which bind via the kinase domain, they have a very small zone of activity because but most of the other proteins which bind to Talin tend to have this this mode of binding where they have a binding site, a Talin binding site, which is separated from the rest of the molecule by a large unstructured region, this spaghetti which everyone sees in AlphaFold. And then the enzymatic domain is at the other end of this. And what that kind of does is it means that each of these enzymes which have got a tether in this way are tethered to a certain location in the cell. So this one is an enzyme we're working on at the minute. This is bound to a domain, and then it's got its unstructured linker, and then that enzyme, enzymatic domain, is free to operate anywhere within this sphere. And this is exactly the same as this goat in a field where he's tethered by this string and this stake, and he'll eat a really tight circle of grass, the length of that rope around that center point of the stake. If you make the rope longer, it'll eat a longer patch of grass. And if you move the stake, it'll eat a different patch of grass. And this is what's happening here. This enzyme is operating here. If you move these closer, it could then reach the membrane and work on membrane substrates. But here it's not going to reach the membrane. So we started to get this idea that maybe these tethered enzymes have got discrete zones of activity which can be moved by moving the tether point. So we went back to AlphaFold, we went back to our structural modeling, and we went back to measuring the distance between these tether points and the enzymatic domain. And that enabled us to draw these zones of activity, the same as here, but for all the other different proteins which are binding to Talin. And then we got to raise this question of what this would look like inside a cell. And obviously, as Claire pointed out, the cell is in culture or cell, it's very noisy. There's a lot of contributions. There's a lot of inputs. If you really want to have mechanical computation to happen on with high fidelity, what you need to detach the mechanical computation into an isolated mechanical unit distal from the cell body. And that's exactly what I think a dendritic spine or a synapse is. You've got this branch, and then you've got this headlight region. So we chose to focus on the synaptic adhesions just because they're a much simpler system and a much simpler geometry. And if you look at the dimensions of these tend to be about 1 micron by 1 micron. So now if we go and draw these to scale on the same way that we've drawn Talin to scale, then what we can see is that Talin is shown in the fully O state and then the fully 1 state here, fully closed and fully open. You can see that Talin easily spans the length of these spines, these mechanical units. And here I've drawn on, we modeled on all 11 vinculins to show where they will dock onto this. So what you can immediately see without is that this Talin, as it opens and extends out, it layers across this unit with these binding sites positioned at fixed locations away from the integrin attachment sites, which have been reported to be around the edge of the postsynaptic density. And now we can take the zones of activity, which I mentioned on the previous slide, all drawn to exactly the same scale, and we can start to lay these onto this. And what you can immediately see is, depending on the switch patterns, you spatially organize these molecules in Z, coming away from the top of this thing, of this spine. So they're moving, as you open these switches, you spatially organize these and separate them out and resolve them in Z. And as these patterns change, you could update and reposition and reorganize these molecules. That's the general idea. And this is something we're desperately trying to prove, because I think that if this is true, then this would really explain a lot, and it would point to this binary coding running a lot of neuronal activity. And what you can see measuring the length of Talin is that they are much longer than at rest. They're stretching to 300-400 nanometers. So coming a sizable distance down each of these spines, all in one direction. But this led to this idea and this thinking about this, that you could kind of imagine that within a spine, but also within any cell, you could imagine that all of the molecules could be separated into 3 sort of types, 2 ordered and 1 disordered. You've got things which are physically attached to membranes or to organelles, which are static relatively. And then you've got things which are freely diffusing. And then you've got another ordered component, which is being physically moved by these switches opening and closing, recruiting to specific locations in Z. So then you can imagine that you could have a scenario where you have a static component and then you move the other layer up and down relative to it by switching on the motor proteins and switching on contractility and reorganizing these synaptic scaffolds, which don't have to just include Talin. There's other synaptic scaffolds are available, but Talin is the only one which has got these 13 binary switches in order. But then you can start to imagine that you could look at a dendrite, which has got 100 or 1,000 spines on it, and each of those spines has got different activity. And if you look with a microscope, they all look exactly the same. But here you could imagine that by spatially organizing these enzymes relative to each other, then some of these combinations might give rise to high activity where all the enzymes are lined up, so you go and some they might be held apart, which means that they'd have suppressed activity. And this would be an idea we'd think about where a signal coming through the synapse, switching on the motor proteins, would pull specifically on the one it was stimulated, because that's where the motor proteins are switched on. And that's experimentally shown by others. But here that switching on of the motors would pull on these scaffolds and update these patterns. So the analogy which we use, which is really nice over this side of the pond, but isn't good for all the ones in America, because you tend to have automatic transmission. But in my car, we've got a manual gear stick. And the way that a gear stick works is it moves the mechanically operated part of the machinery relative to a static part of the machinery. And in doing so, it changes the transmission through that gearbox. And that's kind of what we're trying to test is happening here, where you're changing the transmission by organizing these components relative to each other. And then you'd update this by physically sending signals, which would then change the weight and change the activity of each of these spines independently. And if you could get on board with that you could imagine a mechanical way to dial up or down the activity of a synapse, then by writing information into it, then you could imagine that you could write huge amounts of information in binary format in exactly the same way. You send a signal, you switch on the motor proteins, and you update the register of these switches, which then changes the flow of electricity around that circuit. And if you look at what the cortex looks like, it looks perfectly built for such operations, because it's incredibly logical and it's incredibly repetitive, comprised of 2 million of these cortical columns, which are just a network of neurons. And these teardrops, these little red teardrops, pyramidal neural cells, they can have up to 60,000 different synapses where they're intricately wired to all of the other neurons in this layer. And then they have trunk wiring back to the hippocampus and the memory control center. And what's interesting to me is that this is an arrangement of synapses. If you could imagine you could dial these all up or down as a function of signals, you could physically write information into them. And what's striking to me was, if you look at as we've tried to develop and optimize data storage in silico, so we've now got terabyte disks which will fit on the end of your fingernail. The way that we do that is to have many, many identical repeating channels, each repetitive, they're all exactly the same, but each one given a specific address, so you can write specifically to that site. And then you call it back by the flow of electricity back through upon calling it. And I think that this indicates a way to write information. And just to finish up, this leads to this idea that everything we do, everything we see, listening to this, but everything from the minute we're born, triggers the sensory neurons and send action potentials into this calculation. And it would just tune the weights of all of these synapses by just pushing and pulling on these switches, which may or may not be true, but I think it's extrapolated based on this core principle of these memory molecules, which are being controlled via signals switching on contractility. And I like to think, and I like to argue, and this is philosophy, probably mathematical philosophy, if that's a thing, that you could write from the minute that the brain's developed in utero, it could start to be written with information based on the uniqueness of that animal's life. So it optimizes that individual for its own circumstances. So just to finish and to ground it back, and if I've got one more minute after, so much just one thing quickly. This is quite wacky, but it's also based on experimental data. We can physically show that talin is a memory molecule. We can physically write information into it using small changes of force, and we can read it out by looking what's bound to it. And it's present in every single cell pretty much in our body. And if all of our cells are mechanically synchronized, you could kind of imagine that this is a machine code. All of the cells have got switches all over them. Every single organelle, the nucleus, the cell, they've all got binary switches. So you could start to imagine that this is a binary, we're binary creatures, is what I'd like to put out there. But just going into the brain, I'd just like to say that I reckon that this is the physical location of data storage in animals. You write information into the shapes of the molecules in such a way that you control the flow of electricity. And that sounds wacky, but to me, what I like most about that as an idea is it's completely consistent with all that's known about electrochemical signaling, where the circuits and the electricity goes round. But the difference here would be that the actual information processing and storage would be happening on the mechanical layer under the hood, and it'd be read out by looking at these changes in electrochemical signaling. And because everything's getting converted into a binary format, I think that that indicates the read-write memory system. So I look forward to getting shot down on that. But I think that's basically an extrapolation of what we're physically seeing on these individual memory molecules. And I think that that enables for classical computational information processing systems and some sort of mechanical supercomputer. So I'm going to finish there, but if I'm allowed 2 more minutes, I'll just show something afterwards. But I really want to thank a few people, in particular, Yan Ji in Singapore. This has been brilliant ever since that first experiment, that first email. Every single experiment which we've done since has just opened up this whole new way of looking at things and everything. So it's really exciting, and it's an ongoing, and the stuff we're doing at the minute is just, yeah, it's brilliant. Sam for making these videos, which have been so helpful for showing this. Martin Schwartz and Nick Brown, who have been constantly challenging me and telling me, pulling on the reins a little bit, which has been very helpful. The guys in the lab and everyone else who's collaborated to this. And I just want to finish with this. I did have it in the talk, but I wanted to just flag it here because it came out in this month's Journal of Cell Biology. But just to indicate that we've discovered that there's an extra exon in the talin-1 gene, which has been missed for the last 40 years. And it's really important because it changes the switch pattern which form, because it destabilizes 2 of the switches at the start of the talin rod. And just as for everyone who's working on talin-1, and I assume there's lots of us, it's essential, I think, to check which version of talin-1 your cell type is using, because they behave completely differently and respond to drugs completely differently depending which version of talin they are using. And if you work on pancreatic or skin cells, then talin is exclusively the long form with this new exon in it. So that's just as an FYI, I suppose. And thank you, everyone, for listening.
Thank you so much, Ben. That was amazing. I'm going to digest the fact that I'm a binary creature. Okay, so we have a question from Prabhu, is asking from the initial part of your talk, does the force get transmitted throughout the domains equally? And does this depend on how much the domain would open up in the direction of the force, or the time scale of observation, or the extension rate?
Oh, sorry. So they do get transmitted through equally, and you would predict that the ones at the end would unfold first, but the ones at the start are very stable. R3 unfolds first. But then the other complication, what we're currently trying to do with G is what we've worked out here is the switch patterns we form with pure end-to-end pulling, which is really important. But then that also gets, when a vinculin binds, it provides the next pull is pulling against where that vinculin is. So the order will change as a function of that. So the complexity of this and which switch patterns you end up with is a work in progress. And I don't know if that answered the question, but one of the things we're really keen to do is to measure that, see what states these are in. And we're developing probes now to actually read the shape of these molecules so we can see what we've got.
Okay, fantastic. So we'll just go through these what's in the chat. And also, as usual, if people want to raise their hands and ask their questions themselves, I'll encourage them to do so. So we have another question from Aaron Cram asking if stretching reveals enzymatic cleavage sites.
There are reports of that, yes. And it wouldn't surprise me at all if that's the case. There's a report of a calpain cleavage site in the R10 switch. I've not seen if that was followed up on, but yeah, it would definitely indicate that you could expose binding. You would expect it, because if you imagine you expect if you stretch it 600 nanometers, say, there's a lot of hydrophobic stuff there. There's a lot of conserved regions. There's probably a lot of linear binding sites. So it wouldn't surprise me at all if there's cryptic cleavage sites. There's a lot of cryptic phosphorylation sites. And one of the things which I find most intriguing is if we look at the brain phosphoproteomics or the heart phosphoproteomics, there's phosphorylation of serines and threonines, which normally reside inside the hydrophobic core of these domains. So these domains are mechanosensitive because they've got serines and threonines inside them, which enables them to always refold back to the same state. If you phosphorylate one of those, it will stay locked in the open one state. So these patterns can be made more persistent by enzymatic reactions, chemical changes, phosphorylating them.
If I'm next in the, I think I'm next in the chat.
Yeah, go for
order.
it. Sorry, I was waiting for Ankita, but I think we lost her. So Claire, please go ahead.
Okay, so I said I agree with your drawing in the spine of how Talin is going to stretch along the axis of the spine, because in that case, the actin retrograde flow is going from the spine tip to the base, but it's going to be along the membrane. But it is, I mean, in that dimension, it's going to stretch half the length of a spine, just like you show. So I totally agree with that. But I'm thinking about the idea of binaryism when you have an ensemble of thousands of molecules that are all in different states of, I mean, if you frap Talin in a mature focal adhesion with shitloads of force on it, it's still binding and dissociating on the order of seconds, right? So any one Talin in there is going to be going in there, getting stretched to some amount, and then dissociating from the integrin and the focal adhesion and coming back. So it's like this drawing where everybody is a distance from the membrane. Do you really think that ever happens? I don't think that would ever happen, because it's going to be, I think what you're going to get is like, I think just like with a computer, when you take the higher order sum of lots of binary stuff, you get emergent properties. And I think it's going to give rise to emergent properties that we could never predict, because you have all of these different Talins in all of these different states of stretch. That's going to give some information that you would never imagine from a single Talin. No,
sure. So one of the things on that is, originally a big challenge to the idea was that if you ever explicitly dependent on every bit of a domain of a Talin, then if that Talin pings off and gets turned over, then you corrupt that information. So a much more logical part, and what I tried to show in that machinery moving up and down, is that it's the whole machinery which is moving. So it's
the whole machinery of each Talin. It's not all the Talins in sync.
Each Talin's part of a machinery which is moving this stuff. And the idea for the whole thing came from this idea that you've got gaps and gaps for contractility on the Talin switches, so that even though you might have them in different states, you're going to have a certain amount of contract. I think there's a solution to that, where the positive and negative regulators of contractility even out, so that across the whole focal adhesion, say, no, well, not focal.
There might be an average,
because they're so dynamic that they've tricked everyone into thinking that these are incredibly amorphous structures. Whereas I think there's a higher ordering in these in tissues. But even in there, where they're migrating, there's an ensemble average where all of these interdependencies might balance out to give rise to the net effect.
Yes, I agree, but I don't know that we can. We're not I mean, I love the thinking, Ben. I love it. I thought it was a lovely, lovely talk. And but it's like ChatGPT. We don't ever know what it's going to spit out. You know what I mean? The result of all of those talins in all of those different states,
Yes.
it's going to spit out something that we wouldn't predict, even if we understand the molecule very well, because it's emergent. It's going to have emergent properties.
But it might also be, because of interdependencies, that there might be discrete states within that which are stable. That's the idea, I would say. And I'd say that there's probably not 10,000 activation states of a synapse. Looking in the literature, it looks like there's probably more like 20 to 30, depending on who you believe. And their end states of a calculation, moving this machinery, would be what I would argue, but not everyone, I'm sure, would agree with that, but that would be the idea.
Yeah, anyway, it was really beautiful. Thank you so much for stimulating my brain.
Thank you, Claire.
Thanks for that great discussion, y'all. I'm going to ask a question that we have from one of our YouTube viewers. They're asking, do you have any idea of the force range that's applied to Talin physiologically and how this correlates to actin applied forces? And is this required actin-based stress? Does it require actin-based stress fibers, or do you think branched actin would be sufficient?
So the forces used, there's been multiple groups use tension sensors on Talin and on related molecules. And what you normally see is that the tension remains fairly low with transient increases in tension, and then it drops, and then you get a low contractility. So they're a low tension again. And the idea would be that that increase in contractility happens until you cause an unfolding event, and then that introduces slack into the system, and that then reduces the tension. So then it would be that the motors or the, I think the retrograde flow can activate R3 for sure, because we could activate that just with flow in our chamber when we're trying to measure vinculin binding. If we drew it across too hard, the flow would activate it. So I think there's all of them as possible. And I think cortical actin, sure, I'm sure if it's pulling and exerting tension in the right way, I think it could definitely. And I don't think it needs huge stress fibers to trigger this.
Okay, great. I think that's the crux of the question. From Ajay, are mutations in Talin associated with learning or neural defects?
Yeah, so I don't want to say too much about that, but yes, they are. And we've got a class of a new class of childhood early onset epilepsy, which is caused by mutations in the pulling part of the Talin molecule. So the idea there we're trying to develop is that is a result of the mispositioning of these enzymes as a function of because they're supposed to be pulled to here, but they're only here, and they get out of sync, and then you get a re-entry circuit. So I think that yes, they are mutations in Talin-1 for sure.
So I'll just add, I guess, to that. It was a really, really fantastic talk and a very creative idea that you presented to us. So I mean, the fascinating sort of prediction from the model you've shown is that mutations in different parts of the Talin would possibly give rise to very distinct sort of categories of problems, and that if you did a comparison of mutations in different parts, that there would be kind of almost different syndromes associated with
I appreciate the yeah, as an idea that sounds good, but the problem is that Talins work in every single cell type. So mutations, there's not many mutations in the two Talin genes which are viable. More often than not, they're not. But something which I think is interesting on that is that integrins, there's 24 integrins, and if you get a mutation in an integrin, you get a very specific disease, like if it's involved in clotting or it's involved in the immune system. We have got a mutation in Talin which came from a patient, which was ever so subtle. It caused a tiny, like 5% change in integrin activation. But because it affected the integrin activation, it affected every single integrin activation. So he's got the most broad spectrum, like every single, he's got lots of things, lots of ailments, which indicated all of the things where Talin's essential to me. But it basically had all the, it was like basically having mutations in all of your integrins because every single one of them was slightly defective. But there's not many mutations yet discovered in Talins.
Right. Great. Amazing.
Yeah, thank you.
Ajay, since you're here, do you want to just ask your second question as well?
No, I think it was just a comment. I just liked Claire's idea about the emergent, that she would expect emergent properties based on the collective state of all the different. I thought that was a cool idea.
Yes. And the
Okay,
timescale.
great. Then, yeah, the timescale question. Do you see that, Ben?
Oh,
yeah,
that's right. I forgot about that.
I'm being nosy and looking ahead. Yeah, I think the timescale. I think as soon as the tension's sufficient, a domain unfolds instantaneously. But with all of these cell signaling pathways, and like with growth factor signaling, you get immediate effects and then longer term effects. Once you set the ball in motion of a cell signaling cascade, the timing becomes sort of secondary, because as long as you activate that receptor, which activates that gap, which then activates that whatever and gets contractility switched on, then you triggered that, and then it can pull and change that for future. So the timings are all to be worked out.
Actually, my question was more related to the idea that people have studied the dynamics of spine morphology, and whether in the studies that have concentrated on that, not necessarily how it's linked to the mechanism you describe, whether it's known whether these changes are linked to short-term changes in memory or long-term changes in memory. So it's a more sort of a neuroscience-y question, but I don't know that you know.
The big challenge with all of the neuroscience stuff is because these are perfect sealed mechanical units. The second we detach them and look at them outside, the mechanics, the perfection's gone. They start to just fizzle out and melt away. It's really hard to visualize spines because they don't want to exist until they're in that perfect sealed mechanical unit. But we're trying to collaborate with neuroscientists to look at some of that stuff and to actually link this from a nice idea to a neurological function.
Great, I loved it. I'm going to recommend you to the neuroscience series at the NIH.
Amazing, thank you.
Awesome. All right, so we're definitely going to go past the hour. Are you okay, Ben, to stay on and keep answering questions?
Yes.
Okay, great.
Do it in 20 minutes.
And I guess you owe Dave a I don't know, a coffee or something for getting sick
last night.
Our next question's from Mehmet Sen. Wondering if you've observed refolding of Talin domains after stretching, for instance, in molecular tweezers, and if so, molecular tweezer experiments. If so, have you observed refolding in cells?
Yeah, so they definitely refold with high fidelity, because all those images I showed were 6 or 12 cycles. You can pull and open and close them all day long on their own. Interestingly, if you add in vinculin, and particularly constitutively active vinculin, then you can open it, but if constitutively active vinculin binds, it never lets go, so then it will never refold. And we can measure that, and then that enables you to show with wild type vinculin, that's not what happens. It binds, limits refolding for a period of time, but then it can come off because it will auto-inhibit itself. So the dynamics of refolding is something we're very interesting and is very quantifiable for actually measuring force-dependent binding constants. You can see how quick it takes for these things to refold or to be displaced.
Great,
and
fantastic.
The fact that the cycles of elongation and stretching and contraction seen in contracting cells by Mike Sheets's study, I think that indicates that these are folding and refolding.
Okay, awesome. Our next question is from Bob Fisher, asking about, are there different domains? Doesn't each different domain have a different spring constant? And then wouldn't the unfolding order just sort of be dependent on those different spring constants?
Yeah, they do. I mean, like as when Claire asked me a question at the start, like we could categorize them into different ones, where really weak ones, quite mechanically amenable, pretty strong, and then the ones which are like really solid and probably unlikely to unfold, and that could be distilled out as a spring constant because that's the force threshold at which it would open up, and the order is dependent on that. Yes. The complication there is that with 3 of them which have got very similar spring constants, you can't say for certain which of those 3 will go, certainly not with the resolution we've got in our systems, and that's in the absence of all the other factors. So yes, I think. Okay,
fantastic. Yeah, I think it's a very natural question, right? When we see this opening, is just wondering how how consistent it is. So that makes a lot of sense. So from Hani Suleiman, says, nice talk. Experimentally, is there any evidence to differences in the talon length as the dendritic spine matures from Philopodia to the mushroom-like shape?
That's something we're actively working on. At the minute, no, we don't see. We see differences between different spines, different lengths, but they're presumably mature. It's still very much a work in progress. So you'd envisage it probably would, and you'd envisage that when two neurons touch each other, that as they mechanically synchronize, then you'd end up with specific patterns on both sides of that linkage, would be the idea.
Ben, can you just help me understand? So I'm used to thinking about, sort of crawling, mesenchymal or epithelial cells when you think of a lot of force being exerted, through integrin-mediated focal adhesions and everything. But for neurons, I mean, how mechanically active are these synapses? Is there any sense, because I guess I'm realizing I don't have a great physical understanding compared to crawling cells.
So crawling cell, obviously, you need to generate huge amounts of force, huge amount of traction force to physically move a whole cell from here to here. In a thing which is in a sealed unit, and you've only got to switch on contractility to cause shape changes in proteins which are already wired up, I think the forces can be much lower. I think there's a huge amount of, in a bit of a follow-on to AJ's question, but there's a vast number of diseases, neuro learning difficulties and other things associated with dysregulation of contractility in neurons. So if I think the forces are much lower, and for a long time I was always told that mechanotransduction probably doesn't happen in the brain because it's so soft, but then I actually think that's the strength, because then you can make a mechanically isolated thing, and then the only forces you need is sufficient to cause a transition of a protein domain, which is I'd imagine is much smaller force required than to pull a whole cell from here to here.
Yeah, it's fascinating to think about, and also just what's going on in development mechanically versus, adult just learning and
absolutely,
as we just really sit here with our squishy brains. Sorry. Next question from Olivier Pertz. Do you have a rough idea which different proteins bind to specific talon mechanical states, and which would then which morphogenetic programs they would induce?
So the list's ever growing. Most of the interesting ones which we haven't published yet, I didn't put on that list, but we've got multiple enzymes which are decorating this, and morphogenic thing. So trying to take it back to like the classic work to Dennis Disher and these things, of these stem cells differentiating as a function of stiffness. That to me, that was in my, I don't know where that went. I normally have that in my slide deck. I think that's a beautiful study. But by physically changing the stiffness, that changes the mechanical states in the talon, would be what I would argue. And then you get the global mechanotransduction downstream of that. I would say the talon's sitting at the top of all of this mechanotransduction.
Awesome.
Great. Yeah, Ankita, welcome back.
Yeah. Thank you. Next question is from Artem asking, can a protein be an intermediate state between folded and unfolded for saving information?
So another thing which I've drawn here, which there may be a question lower down, is that I've drawn all these things fully extended because that's the maximum length. Obviously, if it's not under tight, if you hold it really tight, it'll be like that, like a tug of war rope. If we pull really hard, it'll be fully straight. But if we don't pull really hard, it'll probably bow in the middle, so it'll be shorter than that maximum length. If these things have got something sitting on them, or they've got a phosphorylation on them, it could come quite close, but it would still not be able to refold because it couldn't refold because it's got something bound, or it's got a modification which prevents it. In which case, you could have it a much shorter length, like it could just move in and out. So whether there's intermediate states, the good thing is here, I think you can say with quite high certainty that there might be different states once it's open, but it's either folded or it's unfolded. So I think there's two states at that level.
Okay, thank you for detailed answer. I started to think that one of the first computers that were developed, they use not binary systems, they use a triple system. I mean that each has three states, for instance, -1, 0, and 1. And due to this reason, I ask this question. Maybe in biology also, it can be used not binary system, but some another system for saving information.
Yeah, of course. I think in terms of the actual Talin and the switches, I think that they're two-state, but then there's 13 of them. What we don't talk about here is the direct connections to the nucleus and the mechanotransduction, which comes from synchronizing these different systems together. So there you could easily have different higher order states, which are, yeah, so I don't know is the very short answer, but yeah, interesting idea.
Okay, thank you for answer.
I think next question was from Dhanur, if you're around. Wanted to ask the question. Perfect.
Yeah, thanks, Ben, for the talk. So my question is, how does the mutations in Talin bring about aberrant cellular fate specification at the time of neurogenesis?
So I say it again. So how do different Talin states?
No, if there's a mutation in Talin, in Talin protein, do you think that there would be aberrant cell fate specification during neurogenesis?
Yes, definitely. I think if you've got no Talin, then you don't even get past embryonic day 8. If you have got Talin, with mutations in, the fact that there's very few which make it to birth, you probably lose a lot of them by failure at specific morphogenic stages, which don't complete, and then it doesn't continue. The few which get through, which give rise to these neurological diseases, I guess with 2 Talin, there's a bit of redundancy in the system until you need to be fully quantitative, because you've got 2 copies of Talin 1 and 2 copies of Talin 2. So a lot of the broad stroke stuff you could probably compensate, but then specific things which need to be quantitative, then the exact number of them and the exact number of switches and the exact probably comes into play. But it's all to be worked out. Donna.
Thanks.
I think we have a last question from Anna. Says, beautiful talk. And she's curious about what happens to Talin unfolding if FAC is not there, because the binding is important between them.
So if FAC's not there in a fruit fly, then I don't think anything happens. But if FAC's not there in a mammalian cell, then yeah, I guess you get defects in some of these processes.
Yeah, hi.
Hey,
How
Anna.
are you? And good to see you.
Nice to
Yeah,
see
one
you.
of my questions is if you're looking at the binding of FAC. So you presented CDK1, but FAC I think is a main actor during adhesion maturation, and then it's very important for elongation. And that correlates a little bit with the unfolding of Talin. So I was wondering if you see changes in this unfolding of Talin when FAC is recruited, since we know that that's all this mechanochemical transduction of phosphorylation and many other proteins.
Yes. So I think that, yeah, you're right that FAC's really important in this. And I think tyrosine phosphorylation at the early stage of this is really, really important. I think that the interaction between FAC and Talin is complicated. I'm not quite sure how that occurs yet. Some of the things we've tried, we don't see the same as what's being reported. But if you've got a lead on the FAC Talin, someone's giving you nice bunny ears as well, which is
that's Bob.
Yeah, I mean, if you've got a lead for how FAC binds to Talin, which we can do ourselves, so I'd love to test that, because that would be interesting, and that's what we're trying to get back at now.
So you will point that probably FAC is recruited by vinculin?
Possibly.
And then it will be exerting more or less same type of changes, because when these bind together, also they make a lot of changes in recruitment of other proteins, right?
Yeah. No, I mean, I agree. It's just we don't always see, well, we don't really see FAC binding to Talin biochemically, so that limits, because it'd be good to do that on the single molecule level and see what's going on. So
the interaction.
Maybe we should chat offline on that, because I'd love to know if you've got any ways to get at that.
Yeah, I think we have to also wrap up
Thank you.
because Waterman Lab has to go for journal club now.
Oh,
Thank you so much, Ben. I think we're done with the questions. I'm not missing anybody's.
Yeah,
thank you
we're all
so
set on
much. Yeah.
YouTube, so we're good.
awesome. Thank you so much. And
Sorry,
this has
go
been
ahead.
a lot of
Okay.
fun.
And sorry, yeah, we could only tell you last minute about days.
It worked out
Because-
really well, so I'm glad. I hope Dave recovers quickly and it's just a 24-hour clear out thing.
Yeah, I hope so too.
Yeah. Okay.
Thank you so much, Ben. We'll see you around.